Hello, my name is Grace Delpit. I am a biology major at Howard University, and I've had the pleasure of studying this summer in the Bowerly Lab here at Cornell. And I studied the carbon allocation of specifically below ground systems in NAM founders and SLAC1 populations. And so just an overview of the importance of this. So maize corn is one of the most important crops in our world today. And with increasing climate change and increasing drought, it's very much at risk. And so this work kind of looked into seeing how different genotypes of maize react to the drought and then what in turn we can do to counter that. So what we specifically looked at in below ground was root exudates, which is organic molecules that these roots release in a symbiotic relationship with its rhizosphere, so like the surrounding soil and surrounding environment, to help it react to different stresses. And so we did this through well-watered and water deficit experiments. And yeah, so to do this experiment, we were just phenotyping the different plants' responses in these different circumstances, as I said, well-watered and water deficit. And to do this, we use the methods down here, but I'll just kind of go through them to the results. Once we collected the root exudates, we selected, of the 25 NAM founders, 6 specific genotypes for different reasons, but basically we did a large cluster analysis, and these were the genotypes that had the best germination as well as just more positive results. So that's what we chose to look at those. And I specifically looked at total root dry mass as well as surface area scaled and the exudation rate area. And excuse me, these results were all pretty much as expected, specifically with the surface area scaled. As the plants got into the later developmental stages, the surface area increased, which makes sense because the plant is growing. And then with the exudation rate area, it decreased as it got towards higher developmental stages, which is also expected because VE, which is the first developmental stage, is emergence, which is when the plant is growing. So it's doing a lot of communicating with its surrounding environment. So a lot of exudation is occurring. And yeah, the results were very positive, and just confirming what we initially expected with the genotypes and developmental stages having a very large impact on how the specific plant responds to the drought environment. And so moving forward, the next steps is just to further characterize the different carbon allocation in these plants, is putting them in these airtight chambers, putting plants at the latest developmental stage in these airtight chambers to further then do carbon mapping and carbon-13 analysis. So yeah, that's it. Thank you so much. And I would like to say thank you again to my lab and my mentor, Andrea.
Thank you so much, Grace. That was so interesting. You talk about it so with so much knowledge. I'm so impressed. This is so cool. So you mentioned when you're talking about your results, you said most of this was what you expected. Did you get any results that you were not expecting or that were kind of weird? Could you tell me about that?
So the first thing that kind of stood out of the 6 that I looked at was the genotype HP301, which is right here and right here. It just kind of stood out as having a higher exudation rate area and then also total root dry mass. I haven't exactly looked into the why of that yet, but from the graphs that I collected, this one stood out. So it could just be that this plant or this specific genotype just reacts different, and it's something to look forward to looking into in the future.
Great. Thank you so much. Thanks for sharing your work with us. And yeah, thank you.